Hi, thanks for joining us. My name's Nick Ward and this is my colleague Matt Norwood. We're biogeochemists out at the PNNL Marine Sciences Lab. So we're out in the woods just behind our chemistry labs up here, and we're studying greenhouse gas cycling in natural environments. So specifically we're interested in methane, which is a potent greenhouse gas. It has about 30 times the warming potential in the atmosphere compared to CO2, and the global budgets and models are pretty unconstrained in terms of some of these fluxes and mechanisms. We're here looking at actually both uptake and emissions from the soil and emissions from trees. So methane emissions from trees is a pretty recently discovered phenomenon where we're just starting to discover that trees emit significant amounts of methane either from deep down in the soil or actually even created inside the tree. So we've got a couple instruments out here that are field portable. So that gray box over there is a backpack CO2 methane analyzer. So it's measuring fluxes from the soils with that white chamber and the tree in real time. So we can go hit a bunch of trees in this forest. So the other tools we use is to look at what are some of the sources of methane. So is there evidence of microbes in the wood creating that methane? Our tree cores. So we use this borer to drill into the tree and then collect a wood sample from inside that tree. So Matt's going to show you that tree core that we just extracted a few minutes ago. And a lot of people use tree cores traditionally to look at annual growth rates. So you can measure the width of each ring to get that annual growth rate. We're actually looking for either voids inside the tree or rotted out sections. So you see this dark orange section is kind of rotted out, and that could be an indicator of some microbial induced methane production through rotting of the wood. The other tools we have that we're testing out, so this one here is called a resistograph. And this is a resistance drill that's measuring how difficult it is to get into the tree or into the wood. So this is drilling into the tree pretty rapidly, and we can screen for voids that would be indicative of rot and methane production. We also take those tree cores back into the lab and we look for genomic evidence of methanogens inside the wood. So we've actually found microbes inside wood that are creating methane. So with that, thanks for joining our tour.